Welcome to one of the most fascinating topics in biochemistry. So far, you have likely studied enzymes that take a single substrate and turn it into a single product. But in the real world of our cells, things are rarely that simple. Today, we are going to dive into multi-substrate reactions, which are the true workhorses of your metabolism. We will learn how enzymes juggle multiple molecules to sustain life. To understand why this concept matters, imagine you are a barista in a busy coffee shop trying to make a mocha. You cannot make it with just espresso. You also need chocolate syrup and steamed milk to complete the final product. Similarly, most enzymes in your body need to bind two or more substrates together to create a product. In fact, over 60% of all known enzymatic reactions involve multiple substrates interacting with a single enzyme. These are not rare exceptions. They are the standard way your biology operates. Now that we understand what multi-substrate reactions are, let us look at how they actually happen. These reactions are broadly classified into two major categories based on exactly how and when the substrates bind. The first category is the sequential mechanism. In this process, all substrates must bind to the enzyme before any product is released. Because everything comes together at once, the enzyme and its substrates form a massive ternary complex. The key takeaway here is that all the reacting molecules are sitting in the active site at the exact same time. This brings us to the second category, which operates very differently. It is called the ping-pong mechanism, or a double displacement reaction. In this scenario, the enzyme processes one substrate completely before even touching the next. The first substrate binds, a reaction occurs, and a product is released. Only then does the second substrate enter the active site. Because of this strict step-by-step order, the two substrates never actually meet on the enzyme at the same time. This physical separation will have a profound impact on the mathematics we will see shortly. Before we dive into the equations, you might be wondering why we need to classify these reactions so specifically. Understanding whether an enzyme uses a sequential or a ping-pong mechanism is absolutely crucial for developing life-saving pharmaceutical drugs. If we know exactly how and in what order substrates bind, we can design specific inhibitors that block the enzyme at a precise step in its cycle. For example, many drugs that treat bacterial infections or regulate blood pressure work by trapping the enzyme when it is waiting for its second substrate. Without this deep understanding, modern rational drug design would be completely impossible. Now, let us look at the mathematics behind the sequential mechanism. This equation comes from expanding the standard Michaelis-Menten model to account for two molecules binding simultaneously. Let us break down the symbols. The variable V is the initial reaction velocity, and Vmax is the absolute maximum speed. The terms in brackets, A and B, are our substrate concentrations. The Km values represent how tightly each substrate binds. The unique part here is the final term containing Ksa, the dissociation constant. Because both substrates are on the enzyme at the same time, their concentrations multiply together in the denominator of this final cross term. If you increase the concentration of either substrate, this entire term shrinks, speeding up the overall reaction. Now that we have our formula, let us see this with a concrete example from the laboratory. A researcher is studying an enzyme that joins glucose, which we will call substrate A, and ATP, which is substrate B. She measures the reaction velocity at various concentrations of glucose. Then, she repeats this entire experiment at 3 different fixed concentrations of ATP. When she plots the reciprocal of velocity against the reciprocal of glucose concentration, she observes something striking. The three lines intersect at a single point to the left of the y-axis. This visual intersection is the hallmark of a sequential mechanism. But why does this happen mathematically? Let us find out. This visual result we just saw leads us to an important mathematical proof. To understand why the lines intersect, we need to connect the visual shape of the graph back to the equation we learned. In a Lineweaver-Burke plot, we are graphing a straight line, so we must look at the slope and the y-intercept. Our y-axis represents 1 over v, and our x-axis represents 1 over the concentration of A. To find the slope of these lines, we need to isolate all the terms in our sequential equation that are multiplied by 1 over A. This will tell us exactly how the second substrate, B, influences the steepness of the line. Continuing our derivation, we extract the slope formula. The slope equals kma over Vmax plus the cross term divided by Vmax times the concentration of B. This slope equation shows exactly why the lines intersect on the researcher's graph. Notice that the concentration of substrate B is in the denominator of the second term. As we add more of substrate B, that entire second term becomes smaller, which means the overall slope decreases. Because both the slope and the y-intercept change simultaneously as we alter substrate B, the lines will tilt and eventually cross each other. This definitively proves that glucose and ATP must both be in the active site at the same time. Now, let us contrast this with the ping-pong mechanism. Remember, here the enzyme processes one substrate completely before touching the next. Because the two substrates never meet, they do not directly interact. This physical separation simplifies the mathematics significantly. Notice how this formula is identical to the sequential one, except the final complex cross term is completely gone. Because there is no term with both A and B multiplied together in the denominator, changing the concentration of substrate B only changes the y-intercept of the graph. The slope remains entirely unaffected. If the slope never changes, the lines will never intersect. Before we move on, let us see this ping-pong formula in action with a concrete calculation. We are looking at a transaminase enzyme. We are given a maximum velocity of 100 and Michaelis constants of 2 and 4 for substrates A and B. The experiment runs with A at 10 millimolar and B at 20 millimolar. We want to find the initial velocity. Because this is a ping-pong mechanism, we use the simplified equation without the cross term. We substitute our given values into the three terms. The first term is the baseline inverse velocity, while the others account for the binding of each substrate. Simplifying the fractions gives us 0.014. Taking the reciprocal yields approximately 71.43 micromoles per second. This makes physical sense. The enzyme is well saturated, so the velocity is high, but still below the absolute maximum of 100. Now that we have calculated the values and proven the math, let us look at the visual summary of everything we have learned. On the screen, you see two Lineweaver-Burke plots side by side. The y-axis is the reciprocal of velocity, and the x-axis is the reciprocal of substrate A. On the left, we have the sequential mechanism. Notice how the three lines, representing increasing concentrations of substrate B, all intersect at a single point. This is the visual proof of the ternary complex. On the right, we have the ping-pong mechanism. Here, increasing substrate B shifts the line downward, but keeps it perfectly parallel to the others. This parallel nature proves that the substrates never occupy the enzyme at the same time. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that multi-substrate reactions are the standard way enzymes operate in biological systems, not the exception. Second, we classify them into two main types: sequential mechanisms, where all substrates bind together to form a ternary complex, and ping-pong mechanisms, where the enzyme processes one substrate at a time. Finally, you can easily tell them apart in the laboratory. Sequential reactions produce intersecting lines, while ping-pong reactions produce parallel lines. By mastering these concepts, you now have the mathematical and conceptual tools to understand the complex metabolic pathways that keep all living organisms alive. Great job today.